Well, good morning. It is my pleasure this morning to moderate the first session in this room. We have 4 outstanding students who are going to give presentations. All 4 students are students who have done research for me. First up is Andrea Sandoval. Andrea has been doing research for quite a long time, pre-pandemic. Actually, I think she did 1 experiment before the pandemic. So she had just gotten her hands wet, and then we went remote. And she did an awesome job. She worked with other students. She wrote a report that basically literature reviews, summarized everything, and then that led to her getting back in the lab to obviously keep going. And then I also charged her with starting a whole new part of our project, because the project was mainly fluorescence spectroscopy, and asked her to do spectroscopy. So she started that part of the project all by herself. So it's my pleasure to introduce Andrea today to talk about analyzing interactions of calmodulin with viral HIV matrix protein.
Hello, everyone. Thank you for the introduction, Dr. Rue. As you mentioned, I am Andrea Sandoval. I am a biochemistry, and molecular biology major, and today I'll be discussing my research, which is analyzing the interactions of calmodulin with the viral HIV-1 matrix protein. So starting off, we are investigating HIV, which is the human immunodeficiency virus, and it affects over 37 million people worldwide. So it is a very prominent disease that we would like to investigate. And it attacks the CD4 T cell of the immune system, and it could cause autoimmune deficiency syndrome, or AIDS, if left untreated. And until recently, there has been no cure. In the past years, there have been about 4 to 5 cases of complete remission, but that is still a very small percentage of the total population that is affected by this disease. So we are hoping that this research will help close that knowledge gap and eventually provide a more long-term cure for more people. So we are investigating the HIV-1 matrix protein, which has been found in the replication cycle and regulation of the HIV virus. And it participates in RNA targeting to the plasma membrane, incorporation of the envelope into virions, and particle assembly, and it is found in the N-terminus of the GAG protein. And so we are investigating how the HIV-1 matrix protein interacts with calmodulin, and we have found that when calmodulin interacts with the matrix protein, there is conformational changes occurring. And calmodulin has been shown to be upregulated upon HIV infection. So we believe that it plays a potential role in the replication cycle of HIV. And so diving into a little bit more about calmodulin, it is a calcium binding protein found in all eukaryotic cells. And it is activated upon the binding of calcium. And it contains a C terminus and an N terminus, as most amino acids do. And so we will be investigating the full protein and the N terminus and the C terminus. So our study has been focused on determining the secondary structure of this protein and the interactions that occur. And we have also been observing the conformational changes and any in additional interactions. So through circular dichromosome, we aim to determine the alpha helixes and the beta sheets and any additional coils that will help us gain more information about the structure of these proteins and how those conformations change as the proteins interact. So as I mentioned, we are using circular dichromosome. So first, we begin with linearly polarized light, and it is then passed through a polarizer, which then goes through a photoelastic modulator, which converts the linearly polarized light into circularly polarized light, and it is then absorbed by the detector, which measures the ellipticity. So the CD machine measures, is broken up into 2 components of circularly polarized light. There is the left component and the right component. So the left component is counterclockwise and the right is clockwise. And so the protein must be chiral. And to help aid with that. So for example, if your hand is the molecule and your fingers are the different atoms, if you take the mirror image of that and put them on top of each other, they don't line up, so they are non-superimposable, and that is essentially what a chiral molecule is. So the CD machine measures the differential absorption of the left and the right components. And this animation here shows the left and the right components and how it forms a spiral. So our procedure has been a series of titrations. So we begin with either calcium buffer or APO buffer, which has a lack of calcium. And then we add the HIV-1 matrix protein, and then we titrate with calmodulin, either the full protein, the N-terminal, or the C-terminal, depending on what we're testing. And we do that in 0.5 microliter increments up until 4 microliters, because that is double the concentration of what we begin with for the matrix protein. And so our primary results with the fluorescence spectroscopy, which was the first part of this project and is still ongoing, we found that there is binding occurring between the matrix protein and calmodulin, and the full calmodulin in APO and calcium conditions. There is also binding shown with the full matrix protein and the C-terminal of calmodulin in both APO and calcium, and there was binding with the N-terminal in calcium conditions, but there is a lack of binding in APO conditions with the N-terminal calmodulin and the full matrix protein. And so this suggests a possible two-step binding process where the N-terminal binds first and then the C-terminal binds, because as we can see, there's binding occurring with the full protein, but then there is a lack in the N-terminal. So we believe that there could be a two-step process. And so diving into the circular dichromosome data, we have unfortunately not been able to see as much of a change as we would like with once the calmodulin is added. We were expecting to see more of a change with the alpha helical structure, since both of the proteins have alpha helixes. So we expected to see as they bind, they become more compact. And so we believe that there may be an issue with the concentration. We may have to up the amount of calmodulin that is added in these titrations. So we ran the proteins separately, and as you can see with the blue here, that is the matrix protein, and then the red is calmodulin. So you can start to see that alpha helical structure, and then when they are added together, there is a shift occurring, so that means that there is a conformational change. So this more aligns with what we expect to see, and unfortunately we have not been able to see that when we only titrate up to 4 microliters of calmodulin. So we began to run other experiments of upping the concentration of calmodulin. So as you can see here, this one we titrated up to 20 microliters of calmodulin, and you can begin to see that shift that we expected to see, indicating a conformational change occurring. And also with the fluorescence spectroscopy data, there was a occurring, which also indicated that there was a conformational change occurring, and there was a lack of that with the N-terminal. So we were expecting to confirm that data with the circular dichromosome. However, we will need to continue doing experiments with higher concentrations. So to summarize, we saw binding with the full calmodulin protein and the C-terminal of calmodulin protein binding to the full matrix protein in both APO and calcium conditions. And we saw a lack of binding with the N-terminal for calmodulin and the full matrix protein in APO conditions only, and this suggests a possible two-step binding process in which the N-terminal binds and then the C-terminal binds. And so in order to continue this project, we hope to increase the concentration of calmodulin, because since we did not see as much of a conformational change occurring as we expected to. And we will also need to continue this experiment with the N-terminal and the C-terminal of the matrix protein, because up until now we have only been testing the full protein and the separate terminals of calmodulin. And I would just like to thank Dr. LaRue for advising me on this project for the past 4 years and always being supportive of student research. This project is mostly student-driven, so Dr. LaRue just kind of takes a step back and lets us collect our data and then helps us analyze it and help us better understand the bigger picture. And it's been a pleasure and it's helped me really appreciate the field of research. And I'd also like to thank my fellow Cat Lab members, past, previous, and future members who have helped collect this data and kept this project going. I'd also like to thank the Chathuni University Center for Undergraduate Excellence who helped fund this research. And lastly, I'd like to thank my family for being there every step of the way and for always supporting me. Woo.
Thank you. All right, we have time for questions, and I encourage our new colleagues to ask plenty of questions as well. That's Dr. Sandoval.
Thank you. That was great, Andrea. Thank you so
Yeah.
much for your very interesting data. What was the reason to begin with to focus on the N and the C terminus of the matrix protein and not say any other part of the protein? Is there a rationale behind it?
Yes, previous literature has shown that that's where the binding possibly occurs on the ends of the protein, where the binding, that the matrix protein binds on the end of the C terminus to the matrix protein.
So there's no reason, a priori reason to check the middle part, for example.
We test the full protein as well, but the most binding occurs on the two end terminuses.
Thank you.
Questions?
Yes.
I would ask, but I just don't know enough about it.
Other questions? No.
Do you know exactly exactly like what occurs during that binding process, or like what amino acids like interact?
We are not quite sure. We are still trying to better understand why these even interact, what causes there. So hopefully more research down the line will help us confirm which amino acid specifically binds.
Real quick, the concentrations that you tested, are they physiologically normal or meaningful, or would you describe them as being high or low?
So with the concentrations that we chose, we begin with about 50 micromolar concentration of the matrix protein, and the calmodulin is also the same concentration. But in the fluorometer, with the fluorescence spectroscopy data, we have found that about double the concentration of calmodulin shows a conformational change occurring with once it binds to the matrix protein. So that's why we do about double the amount of calmodulin. But unfortunately, with CD, it's a little bit more sensitive, so we can't see those changes occurring as much.
Well, I have one final question then for you. What are your plans after graduation?
Yes, I am planning on taking a gap year before medical school. So during that gap year, I will be volunteering at CHOC. And I'm also currently working at Disney right now at Club 33. So I'll be there in the meantime while I work on med school applications.
All right, great. Thank you. Let's give Andrea another round of applause. All right, well, I'm glad to see that most of you made it over to the new room and we got a nice upgrade out of it. So for our second presenter, I'm pleasure to introduce Quy. Quy has been working on this project for a couple of years, different aspects of it. He is the type of student that is always up for any task he given. So he has worked on many different aspects of this project, and the most recent one was analyzing XPS data about ethanol decomposition. So with that, I will.
No, it's TPD and
Sorry, Kevin's XPS one.
Yeah.
The TPD stuff, TPD and SFG spectroscopy. I apologize. Data that we have collected, and he's done a phenomenal job with this analysis. Really dug in quite deeply, and I can tell you from experience that SFG spectroscopy is not easy to interpret and analyze. So he's done a fantastic job. So I'll hand it over to Quy to talk about it.
Hi, my name is Quy Loi. I'm a chemistry major and math minor. So today I'll be talking about one of my capstone project, which is like studying competing reaction pathway in methanol decomposition under UHV condition using a TPD and SFT. So as we all know, catalysts are very important substances because they are used to increase the rate of the reaction without undergoing like any permanent change to the reaction. And they doing that by lowering the activation energy, which helps create like a more efficient and useful chemical reaction. So here in figure 1, you can see like without the catalyst, you would require like more energy to actually perform the reactions. And so like in industry, catalyst is especially very important since it will help increase the productions and minimizing pollutions, and also like decrease the energy consumptions that was put in. But like as important as catalysts are, we basically do not fully understand the whole fundamental process of the catalytic reactions. So take like for example, we know that for step-side surfaces, they generally are more effective in breaking down like strong bond, like stable bonds. So like in the case of methanol on platinum, step-side platinum would generally breaking like OH bond, so like the stronger bond in methanol, like and while like other surfaces like flat surfaces would break the weaker bond like CO or like CH bond. But in reality, we see like the exact opposite. So we want to know like why is that happening. So in order to like study the chemical property and mechanism that control the reactions, we can like study the decomposition of methanol on platinum under UHV conditions to actually look closely at the reaction and be able to see how the phenomenon works. And for this research, the goal was to actually create a foundation that can be used to understand the reactions, but also to help develop a better and more useful catalyst in the future, which can help to solve or current and possibly future global issues. And we are using like the UHV chamber to actually study the decomposition of methanol. And UHV basically ultra high vacuum. So the UHV chamber will create a UHV condition where the environment is molecularly clean. So the pressure is going to be extremely low, 10 to the negative torr. So there will be like no contamination at all, and we can be more efficient and like in studying the reactions, since the reaction is also isolated. And the UHV chamber also consists of various surface science techniques such as the ion gun, the mass spec, the low energy electron diffraction, and the ultra electron spectroscopy. And ion gun basically was used to clean the sample, and like they're mostly used to clean the unwanted molecules like on the surface. And the mass spec is used to determine like the reaction products. Low energy basically determine like the long range structure of the surface. And AES, ultra electron spectroscopy, are used to determine the quantity of carbon and the oxygens on the surface when it was heated. And for this project, we mostly focus on the two main surface science technique, which is like the TPD and the SFG. Oh, and by the way, here's the USB chamber that we have here at Chapman. It was actually built and possibly ready to work like in the future. So here we have the TPD. So TPD is basically temperature programmed desorptions. It's a method that measures gas product released at different temperature, and we will be able to use it data to determine like the reaction energy and the gas product distributions. It will also give us information about the desorption temperature, the rate of the reaction product, and we can use that to calculate the like predict the reaction mechanism and calculating the reaction kinetics. And the graph is it's going to give us a graph intensity versus temperatures, and we can use it to like analyzing the desorption peak to understand like how the reaction works. Basically, TPD will tell us like what's come off the surface, what molecule come off the surface. And within the USB chamber, TPD will be installed in the sample holder. And there will be like copper wire connecting to the TPD, and the current will run through it in order to control the temperature, the heating up the temperature and stuff. So here, figure 2 is the CAD drawing of the USB chamber. So we have all the component that I said earlier: ion gun, mass spec, AES, LED, and the TPD will be installed like in the sample holder right here. So the reaction will be done inside the USB chamber, and the TPD will monitor the sample like directly. And here we have the SFG, which is the temperature dependent sum frequency generations. Basically, it will measure the change in intensity as the sum frequency when we have like two laser beam at different frequency going directly, like interact directly with the sample, and it will give us like the vibrational modes, orientation of surface species, and we can identify the surface intermediate and their reaction pathway. So basically, the SFG will give us a vibration spectra where we can look at like specific bonds and see how they interact. And the SFG data will be put in a pre-built Python program where we can use to perform the fitting of the data. So the SFG data fitting theory is used to do that, and it will be involved like minimizing the differences between the calculated SFG and the experimental SFG. And there will be parameters where I can change and analyze so that we want to make the fitting as close as possible to the overall trend of the data. So on the figure 3, we have the graph before removing the background noise and after removing the background noise. So removing background noise is one of the process in the fitting, and as you can see on this graph, the fitting was actually doing a good job, but you can see like some of the data was very like out of the fitting, and we have like a lot of noise within the data. And when we actually removing the background noise, we have like a greater like fitting of the overall trend of the graph. And then on figure 4, we have the SFG spectra fit. So before the fitting, we have a lot of noise, so right here. And after fitting, we have a very clean spectra, and the signal is much clearer. So both of the TPD and SFG data that I have is mostly obtained from Stockholm University, since we still the USB chamber in Chapman are still like in the process of troubleshooting. But the data will be analyzed through various methods, mostly like with the fitting, the SFG fitting theory, where we fit the peaks, and they will be comparing with the theoretical results and reference data like from major database. And the combining of these two technique will help us understanding what happened on the platinum surface, like with respect to temperatures. And on figure 5 right here, we have the reaction pathway. This is what expect to happen when methanol decompose on platinum. So on the top figure here, we have the step size. So basically, we expect it to break the OH bonds, and then on the flat surface, it will break like the weaker bond, so CO. And yeah, but we know that like from previous like experiments, like on different surfaces, methanol basically did the opposite. So when analyzing all the data, I was able to obtain like the 4 spectra of the SFG. The two top figure is the CH, and the bottom is the CO. So basically we can see that both the CH is giving the signal like in between the 2800 and 2900 wave number, and this exactly the same with the IR spectra of the CH, where they also appear in the 2800 to the 2900. And the CO is also the same, since you can see they are both around the 2100, and that match with the data that we have. And CO, from analyzing the SFG, we know that the CO bond like breaks between 180 and 200 K, and CO bond breaks between 400 and 420, and CH is between 180 and 200. And from the data, we use those data and compare with the TPD. We know that the CH bond break between 180 and 200 will result in more atomic hydrogen and appearance of C2. And after that, we will see like the remaining C2 dissociate rapidly. This because like when we break this first CH bond, the activation energy barrier will be broken, and the other CH is also dissociate. And we know that from this graph, the OH is going to be dissolved around 160, and that's basically support the breaks of the CH bond at 180. So, and like after the CH bond breaks, there will be like atomic hydrogen on the surface, and it will also breaks, like it's also desorbs actually like at 320. And from this TPD trace, we know that there will be no CO bond breaks, since there is no second peak for the desorption of CO. So CO is gonna have one desorption peak at 420, and there's no more like after that. So we know that it will not break as we predicted on the SFG, and it will, and the CO dissolve between like 450 to 500. And here we have the data that we obtained for the 2023, January 2023 data, where we know that the methanol combustion, like these two, breaks at 160 and 180, while like the oxygen copper platinum is breaks at 190 K. So as a result, like we actually be able to like formulate the whole pathway for the flat surface. So the CH bond will break, like the OH bond will break first when it was on platinum, and then after that is the 3 CH bond, and then the CO bond will desorb like between 400 and 420. We don't know if it's gonna break or not yet, since all of the SFG and TPD doesn't provide enough data for that. And the atomic hydrogen on the surface will then form the H2 and dissolves around like 320 K. And basically we successfully be able to understand the pathway, like the mechanism of the catalytic reactions on like fast platinum surface by doing this. And for future work, we basically want to done this experiment here at Chapman instead of Stockholm University. And for someone who will continue my works, they can do experiment on oxygen copper platinum, possibly TPD and SFG, and they can also do the step side platinum surfaces. And thank you for your attention, and any questions?
We're calling forward.
Yes.
How are you looking to apply this in practice?
Well, actually, for this one, we just want to like understand the fundamental process first, and then people can use this to predict like future experiment where they can develop like catalytics.
Any other questions? All right, well, I'll ask you, what are your plans post-graduation?
Oh, actually, gonna do a master in food science instead. Yeah,
Here at
Chapman. Yep.
All right, so I'd like to introduce our next speaker, Ishaan Shah. Ishaan has been doing research measure for 4 years since the first year. He's been very productive. He has a lot of data and analysis stuff that I'm not show in his presentation. He's also been quite accomplished during his time in my group. He was awarded a National Science Foundation International Research Experience for Students Fellowship, which he then had to decline because he was awarded the prestigious Beckman Scholars Fellowship that same summer. We have both of them in the same summer. Ishaan also is a kind of utility player in my group, meaning that whenever I have an issue or problem on another project, he is always there to step up and help out and get parts of the machine and design parts and so forth. So because of his contribution actually on what we just talked about, he is going to be joining me for 3 weeks this summer in Stockholm to collect some more SFG data. So that I will hand it over to Ishaan to talk his projects on core shell networks.
Yes, thank you, Dr. LaRue, for introducing me. As Dr. LaRue said, my name is Ishaan Shah. I am a Beckman Scholar as well as a chemistry major and with a minor in computational science. And my project is going to be focusing on examining photocatalyzed reactions on the surface of gold core, in particular, transition metal nanoparticles. So As my colleagues described before me, Quy described how catalysts are really important to petroleum industry, as well as developing better automobile engines and producing fuels. And in particular, the petroleum industry, we know, as well as automobile engines, output a lot of CO, carbon monoxide emissions, which cause harmful effects on the environment, leads to a lot of health risks. In particular, I was diagnosed with asthma at an early age, and mostly due to pollution in the environment, and I've struggled with that throughout my life. So this project is really important to me in a way where I can focus on a solution to developing better energetically efficient methods to produce synthetic gas. And essentially what we're focusing on in this process is mitigating CO emissions through CO oxidation and hydrogenation reactions. You can see right here, these are two main chemical reactions that involve taking carbon monoxide and forming CO2, as well as forming methanol. And methanol is actually used mainly in petroleum industries. It's fully hydrogenated to form gasoline and petroleum products, which is very important. However, a lot of the processes that petroleum industries use, that automobile engines use, they utilize catalysts. However, these catalysts still are not energetically efficient methods for producing the petroleum products we need. And my project focuses mainly on core shell nanoparticles, which have a bimetallic structure. You can see right here how you have essentially a core metal overlaid with a different transition metal, and essentially a spherical nature, where we're essentially tuning the geometry and electronic structure of the core nanoparticles to then produce favorable reaction outcomes. So in order to describe what core shell nanoparticles are, I first need to describe why gold nanoparticles are important to our project, as well as how they exhibit what's called a phenomenon called localized surface plasmon resonances. So essentially, if any of you are familiar with chemistry, we can think of elements in a periodic table as having electrons, protons, neutrons, and these subatomic particles compose the main substituents of the elements and is very important in chemical bonding. And metals, however, in particular transition metals in the D block of the periodic table, that large block that you all probably don't know much about, but it's there. These metals have what's called a sea of electrons, which it's a continuum of electronic states, a lot of electrons, which you can envision it as filling a bucket full of electrons. And essentially what we can do with that is excited state photochemistry or chemistry. And these sea of electrons, you can see in figure A, how basically can be excited using UV visible light. And through this excitation process, you have these electrons within their electron clouds oscillating, and these collective oscillations in this oscillating electric and magnetic field is what produces what's called a localized surface plasmon resonance. And why that's important is because the collective oscillations then produce hot electron carriers, or a hotbed of excited electrons, which we can tailor an energy transfer from a higher energy state of a metal nanoparticle, and we can tailor that energy transfer to a higher energy state, our anti-bonding orbital, which is the highest occupied molecular orbital of a reactant surface bond. And this process right here is the process of that energetic transfer to oxygen, and then you have CO and oxygen bonded on the surface. We have UV visible light coming in. We have the excited electrons being produced, that energetic transfer, and then it allows us to destabilize bonds on the surface of our nanoparticles, in particular gold nanoparticles. which essentially allows us to form new bonds with a production of CO2. So this is just the oxidation of carbon monoxide. So core shell nanoparticles are really important in a way where we can have a plasmonic core, essentially produce excited electrons, and then also have a chemically active metal shell, which is allowing us to affect the electronic structure of our plasmonic core nanoparticles, which allows us to basically control the reaction barrier of our chemical reactions. And in particular, I focus on, and I've synthesized and characterized 18 nanometer gold nanoparticles. And I focused on 3 different transition metal shells, which are ruthenium, rhodium, and nickel, which are in the D block. So, of course, we're focusing on nanoparticles, which means we're focusing on a scale of 10 to the minus 9, which is pretty small. So when we're talking about transition metals and the interactions between reactants on the surface, we have to describe what is called the D-band model. And you can see in this diagram here how we have essentially oxygen, CO reacting on a ruthenium surface. And we can have basically those electrons from the ruthenium being deposited into what's called the D band. And essentially we have electrons, atomic electrons in atomic orbitals of oxygen, CO, and we have these basically what happens when 2 atomic orbitals combine, we can create 2 new molecular orbitals. So we have one bonding orbital and another anti-bonding orbital, which is higher in energy. And essentially what we can do is by changing, we can change the location of that anti-bonding orbital relative to the Fermi level, which the Fermi level is the highest energetic level that electron can be can reach at absolute Kelvin or absolute 0 Kelvin. And essentially what we're trying to do is mediate that location between the anti-bonding orbital and Fermi level to then bring it closer to the bonding orbital, which allows us to excite electrons from the bonding orbital into the antibonding. So, we can focus on this diagram here, which shows essentially on core shell nanoparticles, we have this energetic transfer where we have energetic electron vibrational coupling, where essentially those electrons couple to the shell metal D band and then also couple to the reactant surface bond, which allows us to break bonds on the surface. So in order to synthesize my gold and gold ruthenium nanoparticles, I utilize gold 3-chloride trihydrate, which is a compound, hydrate compound, as well as sodium citrate, which is acting as a reducing agent. And this allows us to essentially control the size of our nanoparticles over time. And I focus on using a reflux process, which is a typical process used in pretty much any chemistry lab, where you bring water into the into a water condenser and then outlet the water, as well as heat and stir vigorously in order to essentially allow the solution in the flask to vaporize and then recondense back inside. And I utilized a centrifuge to purify my samples and to remove extraneous material during the synthesis that other than the nanoparticles that is synthesized. And for the ruthenium shell, I utilized ruthenium trichloride as well as sodium citrate to control that size. And I focused on different molar ratios, 1:1, 2:1, 4:1, and 1:2 for these nanoparticles. So in order to describe the data that I obtained, I utilized certain characterization techniques, such as scanning electron microscopy, transmission electron microscopy, and ultraviolet visible spectroscopy. Scanning electron microscopy allows you to obtain the surface morphology of a sample, understand size distribution of particles, understand where those particles are located in the sample relative to each other, as well as their overall size. Transmission electron microscopy differs in scanning electron microscopy, where scanning electron microscopy utilizes scattered electrons from an electron beam, which then are detected by a detector. However, transmission electron microscopy utilizes also an electron beam, but those electrons then go right through the sample rather than just get scattered off. And that allows you to get better resolution of data. And ultraviolet visible spectroscopy is allows you to essentially shine UV visible light through a prism and obtain the location of the plasmon resonance and its electronic structure. So these are some of the data that I obtained for the ruthenium and the gold. You can see in the gold nanoparticles that I utilized energy dispersive X-ray spectroscopy, which is a technique focused on using excited X-rays to obtain the elemental composition of particles. And you can see that the EDS, or the energy dispersive X-ray spectroscopy obtained for the gold nanoparticles identify gold on those particles. And the particles have high monodispersity, which means that they're very separated, like they have a lot of space between each other, and they're not typically like combined together. And this is good for our samples for our synthesis of the core shell nanoparticles, which allows us to have a basis of understanding of what our nanoparticles are. And you can see the SEM images for 1:1, 2:1, 4:1, and 1:2. And you can see in particular how the size varies with the increase of either gold as well as the increase of ruthenium. So you can see with the increase of gold, you can see that the size changes about like 10 nanometers. And you can also see the overall composition is around 1:1 here and 2:1 here. And you can see like some of the particles are a little bit more larger than the others, and that has to do with particle aggregation, and particles want to typically aggregate, form larger particles over time. And through analysis, we've understood that our nanoparticles have around 1 year shelf life. And you can see 4:1 have even larger particles, and also 1:2, which were actually a lot closer to 1:1. I expected these particles to be a lot larger, were around 32 to 35 nanometers. So this diagram shows EDS elements present in the sample. And you can see for the 2:1, which there's more gold, you can see how there's not as much ruthenium as here. It's a little hard to see, but there's yellow spots and then there's red spots, which indicate the gold present as well as the ruthenium present. So you can see how the ruthenium changes with the amount present. So I also focused on synthesizing gold and rhodium nanoparticles, which utilized a different method. It involved utilizing olelamine, which is an oil-based organic solvent and reducing agent, which means that the gold nanoparticles were synthesized in rhodium, while as for the ruthenium samples, they were synthesized in nanopure water. And I utilized two different components, CTAC and TOPO, and olelamine to help reduce the nanoparticles. And I utilized different ratios as well. So this is some of the data that I obtained. You can see this is for the 1:1 sample, and I obtained a lot more larger particles, around 45 nanometers. And however, the EDS data showed that the samples were roughly 1:1 compared to the large sample overall, as well as individual particles. So finally, I synthesized nickel nanoparticles, and in order to do the synthesis, there wasn't so much background research on this topic, so I did all the calculations pretty much myself. In the future, we're planning to synthesize different molar ratios, although we only did 1:1. So on first synthesis, these particles were actually quite large, which has to do with the amount of nickel acetylacetate and gold used. And in the future, I want to mediate the different amounts of gold and nickel to establish a different, better basis on the size of these nanoparticles. So finally, I utilize UV vis, and this is absorption versus wavelength. And you can see how essentially the electronic structure changes for the gold nanoparticles, which absorb at around 522 nanometers, and how the core shell nanoparticles no longer have that plasma resonance, which is a good indication that our shell structure is coating the core. And also you can see how the absorbance of the different samples increases as you increase the size of different molar amounts. And you can see a similar process for the oleolamine synthesis as you see in the sodium citrate and nanopure water synthesis. So one of the main things I've been focusing on this semester is building a photoreactor chamber. And this includes having our main reactor chamber over here, which allows us to deposit our samples. We have a fiber optic setup, which focuses our UV light source onto our nanoparticles so we can excite the surface and produce those excited electrons. We have a gas line system and gauges and pressure meters to moderate the amount of oxygen and CO that goes through into our flow meters, which we can then control the actual flow and pressure of those gases. We also have our residual gas analyzer, our mass spectrometer, to determine how the different chemical compounds change as we do a reaction. So as I said, with future work, we still need to fully characterize the gold, rhodium, and gold nickel nanoparticles, as well as use a technique called surface enhanced Raman spectroscopy, which utilizes an inelastic scattering process, where photons are scattered onto the surface, and then basically allows us to determine the vibrational modes and how a molecule actually vibrates. So it gives us a structural fingerprint of our molecules, as well as mass spectrometry to understand how photocatalytically efficient our nanoparticles are. So I want to thank everyone who helped me with the research, especially Dr. LaRue, Dr. Benji Zhang, and as well as Dr. Mala, which helped me utilize the SEM and train me in using this SEM, as well as other students that have helped with the project. I want to thank Summer Undergrad Research Fellowship, as well as the Beckman Foundation for funding my research, as well as IMRI in helping us obtain our TEF data. So thank you.
We have time for one or two questions.
Yeah. So gold has been used in the petroleum industry a lot as a plasmatic metal. There are other plasmatic metals such as silver and copper, which can be used as well. Gold was just what I focused on in particular. And my other colleagues also focus on a different metal called titanium nitride, which is a different core metal and a compound, which is also a plasmonic metal. So gold was just the plasmonic metal I was focused on, but you can also focus on different ones as well.
Another question. All right, and then what are your plans on graduation?
Yeah, so after I graduate, I'm planning to take a gap year. I'm actually going to be teaching a PChem lab for a year. and hopefully applying to grad school in the future.
Great, thank you, Sean, again.
Yes.
All right, so for the last speaker of this section, it's my pleasure to introduce Kevin Alvarado. If I'm lucky, most students I'll meet in the first year, and I kind of have to bring them into my group and get them started early. Kevin, I met in high school. He was still a high school student. He was the very first Simon Scholar student that I mentored many years ago. And when he came to Chapman, I pretty much contacted him and tried to get him right away to join my group. He's been working on this project pretty much the entire time he's been here at Chapman. He has touched every single aspect of it, whether it's programming motors, putting the chamber together, pretty much anything that testing new equipment as it arrives and so forth. So it's a pretty big project. And then this last year, I kind of had him do a side tour on this and actually analyze XPS data from SSRL that we took last year. So he moved away from the chamber and into the data analysis with SSRL, which we'll be talking about today.
All right, so as Dr. earlier gave like an amazing introduction, my name is Kevin, and obviously I'm going to be talking about methanol decomposition on a platinum surface, and this is going to be characterized by X-ray spectroscopy. So obviously, like the main purpose of this is going to get like some foundations for building a catalyst. So for like the third time, you're going to get an introduction on catalysts. So as you guys know, catalysts are very important. They're everywhere. They're in our bodies. You see them, like you hear about them everywhere. And how do they work? So as you see from like the black line, you see this reaction takes like a pathway that requires a lot of energy. But if you apply a catalyst, it actually requires less energy. And this activation energy very lowers. And this is pretty important because it helps the reaction go into like a more energetically favorable pathway. And it just takes up less energy. And what's also pretty interesting about this is that sometimes these pathways that it can take can be like an alternate pathway that can be more efficient. So, I mean, you know what a catalytic converter is? People are stealing these nowadays. And before these were invented, the gas engines, the car engines would be releasing like nitrogen oxide, carbon monoxide, and HT into the atmosphere. And these were pretty harmful. Like if you look back into pictures in LA, like in the 1970s, you're gonna see how polluted it was. But when the catalytic converter was invented, it was able to convert these gases into like CO2, H2O, and N2. Like CO2 is bad, but it's obviously not as bad as it used to be. And the catalysts are also found in like agriculture and like food production and stuff. So that's why I put like a cute picture of a goat. And now, obviously, for this project, I'm focusing on methanol decomposition. And why are we picking methanol decomposition? Well, we're focusing for this particular project on the Fischer-Tropsch mechanism. And this mechanism basically is a way to synthesize hydrocarbons by mixing like syngases and like CO and H2. And in this process, methanol is an intermediate. So if we were to focus on the reverse reaction, instead of going towards the hydrocarbons, and we focus towards how methanol can decompose into these reactants, we want to see what would be like a more favorable path for the products to kind of like decompose, and maybe find like something that like for the hydrocarbons that can be harmful for the environment when they're used up. Maybe we can find something that's not as harmful as it is now, which is like the climate change problem that we're dealing with. And for our catalyst, we're using platinum. And platinum has been a pretty interesting catalyst because as like we mentioned, like some surface science studies have shown that like on the like platinum 111, if this wasn't platinum, it would just be like a flat surface. And if you bring methanol onto the surface, this surface would theoretically be more likely to break like CH bonds on the methanol. And like the terrace surface, the step site surface, which is 221, would be more likely to break the CO bonds in the methanol because they're more stable. But on platinum, research has shown that it's actually the opposite. And platinum 111 is more likely to break the CO bonds, whereas platinum 221 is more likely to break the CH bonds. And we don't exactly know why. So this is why we're doing the experiment, because we want to see what mechanisms are happening and be able to see what is this interesting phenomenon that we're seeing. And obviously, we're going to be doing this experiment on 3 different surfaces. But for this particular one, we're utilizing platinum 111. So to characterize this experiment, the data analysis was done using X-ray photoelectron spectroscopy. And it's a pretty cool technique. It's pretty simple. You pretty much have your sample, which is this right here. And then you irradiate the sample with an X-ray. And whatever is on the surface, as you see here, like the darker blue region, that's what's going to be excited with the X-ray. And then this is the atoms that are on the surface are going to be emitting an electron from like the 1s orbital. And then these photoelectrons are what we measure with like an X-ray analyzing machine. And then these photoelectrons, we can extract the binding energies from the atoms that are present on the surface. And then from this, we can kind of like assign what the binding energies are to each of the elements on the periodic table, and then kind of like figure out the bonds and composition of what's on the surface. So by doing this, when we're reacting our methanol sample on the surface, as the reaction progresses, we can take a lot of measurements of the surface of the sample. And over time, we can kind of see what has been found on like a specific time frame on the sample. That's pretty much what we did. So the method was a little intense. So I just kind of like summarized it. The data was collected at SSRL, which is at Stanford. And basically this place is just like, it's a little hard to see in the background picture, but it's like a giant, like underground tunnel, like a really giant spiral tube. And this experiment was on like over 5 consecutive days. And each run and like all the scans that were collected, they were taken between like 300 to 600 Kelvin, and the pressure was varied between like 0.01 torr to 0.08 torr. And this was pretty different because in like what we're going to be doing here at Chapman is going to be like a 10 to the negative 8 torr or higher, lower. So we should be able to get some interesting information. And then once the data was collected, it was all put through Python, and we were able to analyze like the carbon 1s peaks and oxygen 1s peaks. So this was like the raw data. It looks pretty intense, but I'll be going into a little more detail. So if you look to the left, that's like the intensity, which is kind of like relative to like the carbon and oxygen that was collected on the surface. And that's like, those were all the data sets for like the different days of the week that the data was collected for all the scans. And those are all the temperatures that like the exact temperatures that were used. And then at the bottom, you see all the binding energies. And like you can see there's like a lot of peaks, and you can kind of see like the peaks were kind of shifting over time. And this is what I'm going to be analyzing in a little bit. So for the O1s, this is also like the same thing. We see like the temperatures and the binding energies, which are like around the oxygen region. And like one thing to note is that for the O1s peaks and like the carbon 1s, is that for the carbon containing species, the intensities were significantly lower, like relative to the oxygen. So I'll be going into a little more detail about that. So this was pretty much like the breakdown of all the peaks that we see previously. So if you look at the bottom, like the blue graphs are representing the carbon monoxide that was found on the surface. Then the green peaks represent the water. And then the purple one was just like a platinum dioxide peak that was seen. And I think that just kind of like, because we were at really high temperatures at that point, I think the oxygen that was being released from the methanol was probably just reacting with the platinum, and it just kind of like absorbed into the surface. So that's what we were seeing there. And then for the carbon 1s peaks, this is what I found. There was like there's less peaks because some of the peaks, as you see like back into the carbon 1s, some of them were like all the way to the bottom. So we couldn't really get any information from that. So that's why there's less peaks here. And the red ones represent like CC bonds that were formed during the reaction. The yellow ones represent CH bonds, and the black ones kind of like, it's just like atomic carbon that was found on the surface. So from this, it kind of like led me to conclude that at the lower temperatures, when we analyzed like the oxygen containing species and the carbon, we see that there was like a lot of like C bonds, CH bonds, and CO molecules. And this but then at higher temperatures, there was like more atomic carbon, water, and platinum dioxide. So this kind of like led me to kind of conclude that at lower temperatures, the platinum 111 is catalyzing the oxidation of methanol by like dehydrogenation to formaldehyde, which kind of led to the absorption of like CO and CH. So if you see here, this was like the bond breaks, and then we get like this molecule, and then we get to here. And I think like probably one of the hydrogens kind of like reacted with this, and we got like formaldehyde on the surface. And that's why we were kind of seeing a lot of CO bonds and CH bonds. And then for the CC bonds, I think that when it got to this step, we got like a formate ion, and it probably reacted with one of the methanols. And that's why we were seeing like some CC bonds on there. And for the second part, we see kind of like at higher temperatures, there was like a lot of, there wasn't that much CO. So I think that like the CO bond was breaking at that point. And this is kind of like what we were expecting in a way. And that reaction can be kind of seen here at the bottom, where the methanol reacts with the surface, and then the CO bond breaks. And then we get like a lot of atomic carbon absorbed into the surface. So this experiment is pretty much like a small piece to the like overall thing that we're doing here. Because as you can see, like in the on this image, we're only analyzing what's on the surface, like the elements that are on the surface, like these, but we're not actually measuring what's being desorbed from the surface, or like what the structure looks like. So for the future, we want to conduct these 3 experiments here at Chapman. And we want to do like low energy electron diffraction, which is going to give us like a 2D picture of the actual platinum structure. And we want to do OG electron spectroscopy, which is pretty much XPS, which I was talking about. But this one can be applied in like in ultra high vacuum conditions, which is going to be like 10 to the negative 10 torr. And then thermal programmed desorption, which is going to be pretty important because this is going to give us the opportunity to analyze the molecules that are being released from the surface. So in conclusion, a low temperature platinum catalyst is able to favor the dehydrogenation of methanol, whereas in high temperatures, it kind of like favors the breakage of the CO bond. And then combining our findings from the XP data to what we'll be doing here at Chapman, and also doing it on platinum like 531 and 221, we should be able to get like a holistic mechanism for methanol decomposing on platinum and all these different surfaces, and then aid in like future catalyst design in industrial chemistry. So just like to close off, I just want to give like a big thank you to all my cat lab mates. They've been amazing. It's always nice when I'm just like in the lab, just like feeling stressed out, and they just like come in, and it's always nice to have them there. And they've been like super supportive, like Ishaan and Quy. They've always been there, like even though they're working on different parts of the project, I always feel like really safe, kind of like going there to ask questions. And even though sometimes I don't know, they've always been there to help me. And Dr. LaRue too, because I've known him since high school, and he's been like a great mentor. He's always been there for like everything, like school, like personal life, like everything. And I'm getting kind of teary because it's yeah. So and thank you to Chapman University. Like they're, if it wasn't for them, I wouldn't be here. So yeah, that's my presentation. Any questions? Oh, so like I was talking like with XPS, you're exciting like the electrons from like, you're like removing electrons from like the 1s orbital. So when the electrons are removed, we get the binding energy of that electron. And the binding energy is just like how strong the electrons are being held by that atom. And obviously different elements all have different like numbers of electrons and different numbers of orbitals. So all the binding energies are different. And we can basically, like, if you do a lot of literature search, you can see like all these binding energies have already pretty much been found. So like for oxygen and carbon, which is what we did here. And so just by doing literature search, you can see like the exact binding energies. It's kind of like IR spectroscopy, where like the peaks that are seen, you can just like put them together to like what it would, like what kind of stretch it would be.
The one thing I forgot to mention is Kevin will also be joining the for 3 weeks in Stockholm this summer with Ishaan. You're going to FFG data as well. And Kevin, what are your plans up for graduation?
Oh, well, I'm taking a gap year too, because I'm still kind of like deciding between if I want to do like the medical or chemistry, and I gotta get a job because I gotta pay the bills. So
All right, let's thank Kevin and also all the speakers.
Thank you for joining us though in our second session. Today, I know that we left off.
I have a gift. Oh, that's fine.
Our next speaker is Ricardo Espinoza Hernandez. Ricardo's been in my lab for, I don't know, 2-3 years. Is it 2-3 years?
3 years, I think.
3 years. He's a sophomore, and he has done a fantastic job in taking the lead on this project, a brand new project for our lab on fireflies. I'll tell you all about this, but he has been just such a great leader in the lab. Always troubleshooting, protocoling. You know, sometimes it's a little bit difficult to get undergrads to write stuff down as they do it. We just have some sort of research, but he's done such a great job of getting this project ready to hand off. You'll see the progress that we've made is not quite it's in the form of the question. I won't spoil it, but he's been doing such a great job that I'm really excited to hear about his work. So he's going to talk about the morphological variation in beetle anatomy that contribute to reproductive signaling. So take it away.
Thank you for the introduction, Dr. Waldrop. So have any of you ever seen a firefly? Yeah? All right. So I'm sure we've all recognized them with their kind of flashing patterns of light that they emit that we can usually see at night, and it looks pretty cool. And this pattern of light that they actually emit is a shared ancestral trait in fireflies, which started off as a nocturnal warning signal, but soon adults began to pick it up and use it for sexual signaling, which was different from what beetles would commonly use it, or how beetles would kind of, you know, signal or use sexual signaling, which was in chemicals. And yeah, so over time, there was a reversal from the visual ancestral state to a chemical kind of mode of sexual signaling. And we could see this with this kind of little tree that we have here. That was basically just all the species that we use in our study. And so yeah, so this species, or this kind of tree, is just a small part of this kind of huge beetle phylogenetic tree. And what I want to point out is that you can see in these kind of species that are in the color red, which are chemical signalers, you can see that there's a kind of reversal to this chemical mode of signaling. And not only here, but also right here. And a question that kind of that we kind of made was, if there's this reversal to chemical signaling, is there also or is there going to be a difference in the morphology of the antenna in these fireflies? Because you would think that, you know, they're performing a different function, so they might need to use kind of different tools or different machinery to obviously to be able to perform this. And the first prediction that we made in our study was that the olfactory sensilla of chemical signalers will be larger and wider than those of visual signalers. And the second prediction that we made was that the density, number, and fraction of olfactory sensilla in chemical signalers will be greater than in visual signalers. And to basically answer this, or to look further into these predictions, we decided to focus mostly on a kind of certain part of the antenna, which is the flagellum, as you can see here. And this was basically like the premise of our kind of data collection, because to basically start it off, we had 15 species of fireflies that were loaned to us by Dr. Brown at the LA County Museum of Natural History. And what we basically did was isolate an antenna from our samples and place it under a scanning electron microscope and zoom in very like far into this antenna and produce these pretty cool images that helped us not only measure the lengths and the widths of our cincilla, but also to characterize them between if they were either olfactory or mechano cincilla. And these are basically just some of the images that we were able to produce. And what I kind of want to show here is that we have our samples that were either chemical or visual, and then we also have a sample that was actually both. But what I want to point out here, or what I hope that you can gain from this, is that it's kind of hard to really tell the difference between these two types of signalers based off of these photos. And this was kind of a pretty important piece to our study, because we wanted to kind of show that this is something that wasn't going to be able to be done just by looking at certain photos and, you know, just being like, oh, you know, this is different from this, and that's it. What we had to do was actually take lots and lots of measurements and also perform a lot of statistical tests as well. And going back to our first prediction, which was that the olfactory cincilla of chemical signalers will be larger and wider than those of visual signalers, what we were able to find was this raw data here. And I apologize for kind of like the messed up coloring and stuff, but this one right here is chemical signalers and this one is visual. But basically what I wanted to show was that we collected this raw data that was over 2,544 total observations that we made. And if, you know, the points were here, you could see that there was a lot of consistency among our results. And but we weren't able to actually make any conclusions based off of this, just because these results were from multiple samples of each of our species. So what we had to do was take the averages using a phylogenetic corrected ANOVA. And with this, we were able to find that there was no significant difference in the olfactory or mechano-cincilla length in our chemical and visual signalers. And also, there was no significant difference in the mechano and olfactory cincillum width in our chemical and visual signalers as well. So we moved on to our second prediction, which was that the density, number, and fraction of olfactory cincilla in our chemical signalers will be greater than in the visual signalers. And to do this, we took pretty similar steps like to our first prediction. We had our species samples, and we also put them, or we actually used the images that we took previously. And what we did was basically just click on a whole bunch of points on our images that really helped us characterize the cincilla here. And we basically just denoted them using different colors. And yeah, so what we were able to find from that was that it's just this raw data here. Again, we made over 8,164 total observations. And there was a good amount of consistency as well, except that we weren't able to make any conclusions as well, just because these were results coming from multiple samples of our species. So again, we had to take the averages using a phylogenetic corrected ANOVA. And after doing that, we were able to find that the density of mechano and olfactory cincilla, there was no significant difference between visual and chemical signalers. And also the number of mechano and olfactory cincilla, there is no significant difference between visual and chemical signalers as well. And we also were able to make this really cool kind of tree that shows the fraction of mechano and olfactory cincilla. And what we found was that there was no difference in the fraction of mechano and olfactory cincilla in our visual and chemical signalers. This was kind of interesting because you can see, or what we thought was that, you know, these groups being chemical signalers, they would have a kind of a greater number of olfactory scintilla, just because, you know, this olfactory scintilla is really useful in capturing odors and pheromones as well, but this just wasn't the case. So both of our predictions just weren't supported. So we just finished. I'm just kidding. We didn't. We kept on going, and we continue to ask more questions. And the next question that we kind of asked was, what if it's not the mechano or olfactory scintilla, but instead it's the two kinds of olfactory scintilla that there are, which are trichoid and basioconic scintilla. The main difference between the two is that trichoid scintilla is specific towards capturing pheromones and just specific to capturing pheromones and receiving them. But basioconic, they're really they're open to capturing not only the odors other than pheromones. And you can also see the difference between the two visually, like in this picture. This kind of yellow arrow right here, it's pointing to a trichoid scintilla, and you can see that it's really sharp and pointy at the top, whereas this blue arrow right here, it's pointing at a basioconic scintilla, and you can see that's a bit more like chunkier and wider. And so what we ended up predicting, taking these two types of scintilla into account, was that the density, number, and fraction of trichoid scintilla in our chemical signalers will be greater than in visual signalers. And this was mainly because the specificity that that the trichoid scintilla have to capturing or receiving pheromones. And so what we were able to find was that there was no significant difference in the number of scintilla, or the number of basioconic and trichoid scintilla in our visual and chemical signalers. There was also no significant difference in the basioconic or the density of the basioconic and trichoid scintilla, and there was also no significant difference in the basioconic or trichoid fraction of, or the fraction of basioconic and trichoid scintilla. So our three predictions were wrong. So we, I mean, it was kind of unexpected. And this was mainly because we thought that, there being a reversal event back to chemical signaling, you would think that this kind of different job that these fireflies have to do would require different machinery or different tools for them to use in the process. But unfortunately, or we just weren't able to see this using our results. But kind of future steps that we intend to take are increasing our sample size, because we did only use a couple of samples that we were able to get loaned to us by the LA County Museum of Natural History. And we also plan on characterizing our species between being male and female and seeing if there could be some sort of difference in the antenna morphology then. And with that, I'd like to acknowledge the Wadrup Lab, Dr. Wadrup. You've been such a huge support these past 3 years, and the guidance and mentorship, I really appreciate it. And Nick, thank you so much for all the help and helping me prepare for this, and Daniel and just the rest of the Wadrup Lab. I'd also like to thank Dr. Brown and the LA County Natural History Museum. Yeah, and Dr. Mullah Islam as well, and Chapman University Imaging Facility for being very generous in allowing us to use their scanning electron microscope. And yeah, thank you.
Because you have fireflies that are using visual cues.
Mhm.
But this, okay, and they revert back to chemical cues, but even for the visual cues, those are for mating and such, right? Do they use chemical cues for other behaviors? The ones that have visual
Yeah, so they use visual, kind of those cues for long range communication, and in this case for sexual communication. But they also use chemicals when it comes to short range communication and like direct contact. So they do still use chemical cues, but
Maybe why they're not so different, because even if they were visual, they still had to use them.
it could be, yeah. Thank you.
Question. What are you doing after you graduate?
So after I graduate, I plan on stepping into industry and hopefully just seeing where that takes me into the future.
Thank you so much.
Thank you.
We just started.
Take a little time. First, okay, great. So our next speaker is Sharon Nguyen. She's going to talk about a very relevant topic. I think everybody in their life has had somebody who's touched by depression. And so it's great to hear that she's working on holistic bioactives that might provide a potential alternatives to conventional pharmaceutical antidepressants. So Sharon is also being advised by Dr. Susan Nguyen. Take it away. Susan's ready.
Thank you so much for the introduction. Hi, everyone. My name is Sharon. I am part of the Biological Sciences department here, and I minor in disability studies too. My research advisor is Dr. Susan Yang, who is right there. And today I will be talking about how holistic bioactives can provide a potential alternative to pharmaceutical conventional antidepressants. Do I just hit this button? I'm gonna just do it like that. Okay, sorry about that. Okay, so our number one goal for our body is always to establish balance, otherwise known as homeostasis. For example, if we are experiencing sadness, we crave the feeling of happiness. But depression disregards this balance and prolongs our feelings of sadness from a day to a week to a month, and sometimes even years. So the primary treatment for depression right now are antidepressants. However, researchers now know that
the use of antidepressants of 3 years or more is actually more detrimental to our health than helpful, such as creating addiction, tolerance buildup, excessive weight gain, failure to other organs, and withdrawal symptoms. This is actually not surprising because Western medicine typically aims to relieve symptoms rather than understanding the person as a whole. So with these alarming insights in mind, I challenge you guys to think of other alternatives. But today I will be talking about holistic medicine. So a couple other stats about depression is that it affects over 9 million people and it accounts for over 700,000 deaths per year. So holistic medicine relies on natural properties and herbs to reduce these residual effects and tolerance buildup without actually decreasing and deteriorating our body's insides. So this leads us to propose this question: can holistic alternatives be as effective as conventional medicine? So before the medical revolution, holistic medicine was actually the primary treatment for curing illnesses. So the use of organic herbs aimed to treat the underlying causes of diseases while unlocking our body's natural healing properties. So we actually partnered with a university in Korea called Kyunghee University, and they have this astounding MD program that focuses actually just in holistic medicine. They have hospitals that pair and combines Western medicine and herbal medicine, and our study here further elaborates on that. So we chose 2 bioactives: fermented placenta porcine, shortened as FPP, and Bulpurium reduxi, shortened as BR. So FPP is extracted from pig placenta and has the effects of decreasing inflammation and stress-induced immobilization, while Bulpurium reduxi, BR, is extracted from the plant chow hu. And I apologize if I butchered that word, but it also has antidepressant effects, anti-inflammation effects, and antioxidant effects, both on the molecular and behavioral level. So in terms of methodology, we use female mice subjects in trials regarding FPP because FPP is most notably used for menopausal depression. And for all other trials regarding BR, we use male rat subjects. So with that in mind, we had 4 treatment groups. So the first treatment group is noted as the sham group, and that represents our normal happy mice. Our second group is our stressed mice group, and we actually surgically removed the ovaries of these mice, which causes this physiological stress. Our third group is our stress mice treated with antidepressants, so our pharmaceuticals, such as estradiol or aminodiazole. And our fourth and our last group is our stress mice treated with our holistic bioactives, so either FPP or BR. And after treatment, each of these subjects were introduced to 2 stressful situations. The first being the mice being put underwater for 5 minutes. No rats or mice died, just a FYI. And just like us, if we were thrown into a pool, we would try to stay afloat, right? And this behavior is called escaping behavior. But on the other hand, when mice get tired, they start to give up. They start to exhibit this lack of will to live and results in like an immobile behavior. Similarly, in our tail suspension test, we hung the mice up by its tail upside down, and they also exhibited this escaping behavior, trying to land back on all 4 legs or feet. And they also tried and exhibited this immobile behavior too when they started to give up and lose like a lack of will to live. So when we talk about our results, we see that in our y-axis, we're measuring the total immobility time in seconds. And in our x-axis, we are showing 4 different treatment groups. So starting on the very left for you guys, we have our sham group, which is our normal happy mice. our red bars represents our stress group where we surgically remove their ovaries. Our blue group represents our antidepressants, which in regards to FPP, we treated our mice with estradiol. And our green bars are our bioactive or holistic bioactive groups. So in this case, FPP. So there are a few things that are worth noting about these graphs. First, we see that our controls, our white, red, and blue bars are working. So there is high significance in increasing immobility time when we physically or like physiologically stress out the rats or the mice. And we see that there is significance in our antidepressants when reducing immobility time in compared to our just stressed mice. Another thing that we see is that there is a decreasing trend. However, after statistical testing, we only found that 1500 at a dosage of 1500 mg/kg of FPP, we have some significance, but not for anything less than that. So similarly, when we talk about our BR trials, we see that our controls are working too. We see high immobility time for our stress mice indicated in red. And we see that our pharmaceutical antidepressants, imidazole, has a significant decrease in immobility time. But unlike FPP, after statistical testing, we see that BR has significance in our doses of 200 and 400 mg/kg in reducing immobility time. So out of this primitive study, I want to point out three main takeaways. So the first being that although holistic bioactives were not as effective as our pharmaceutical medicine, it still showed some promising effects in terms of antidepressant effects. Secondly, our study isn't aimed to replace Western medicine or replace antidepressants. It's more so a suggestion to a more sustainable alternative to treating depression. And since this study is very primitive in terms of FPP and BR, we hope to continue adding a computational aspect and regarding the long-term effects and hopefully different dosages between FPP and BR. And I also would like to acknowledge my faculty advisor, Dr. Susan Yang, for giving me this opportunity and guiding me throughout these past 3 years. I also like to acknowledge Kyung-hee University and specifically Dr. Insup Shim for allowing me to visit their lab and collaborating with us. And also I would like to acknowledge Dean Michael Eva for supporting my research endeavors throughout this class. Thank you so much.
Can you hear me?
Yes.
I was just curious as to, I might have been 1 minute late and maybe I missed this, but how is it that an overactinate is a stressor? Is it because it's a surgery or how does it cause stress?
Yeah, so in terms of females, our ovaries have a lot of hormones and removing those ovaries causes like an imbalance in our system, so it causes our physiological stress, causes a physiological stress response. Does that make sense?
So the test routes that you're trying to see if we could use alternative antidepressants to apply to people of different genders, species, and age. So for the rats, I noticed you only use male and female.
Yeah.
Did you do any consideration towards the age or the species of the rats?
We didn't do any consideration of the age quite yet. Hopefully in future studies, we can kind of account for that and make further predictions. We did use female mice, which is a different species, and male rats. So it was like different genders and species. Thank you.
Hey, can you hear me?
Yes.
Okay, so I wanted to speak a little bit more on depression in today's society. Most of it is brought on by a lot of cultural values or things that happen in the environment. And so these things lead to a feeling of like learned helplessness where they feel as though they can't really help themselves. But when you did your experiment, you did it based off of like physiological changes in the body rather than something that kind of builds up over time because of their environment that harms them. So was this something that you guys thought that maybe this was a route you guys wanted to take, or was it just based off of finding a way to stress out the mice and create like a physiological imbalance in order to bring on these medications to try to see if it would be a change or like a difference between that and antidepressants?
Yeah, so of my understanding of your question, like we did like physiologically like stress them out. We didn't really account for this study in terms of like external stresses, like learned helplessness, environmental and cultural things. Because this is such like a very like primitive study to our actual goal, we hope to incorporate those other factors and like test more behavioral aspects, stress them out in a different way, and see if our stress bar kind of is consistent over time. So we hope to do that in the future. Does that answer your question? Okay.
So I believe earlier in the study, you did mention that a lot of Western medicine has specific side effects. So I was wondering if you did look into like the side effects of the bioactive compounds that you were testing as well.
Yeah, so since this was like a collaborative effort, and we did base a lot of our studies from past findings. So we do know that there are like anti-inflammatory, immobilization, antioxidant effects to it. In terms of the long-term effects, that's still our knowledge gap that we still are kind of looking to fill. So hopefully in the future with these data, we can better predict that. But from my personal knowledge, I don't really know the long-term effects in terms of using like these holistic bioactives.
Okay, so I have. Okay. I just wanted to know, what are you doing?
So my plan is to have to obtain a master's and hopefully still pursue the medical school route. My master's is primarily focused in like the computational world and the research world and getting more of a foundation in research.
So since you're gonna go to medical school, which means you're gonna get into Western medicine field, right? How do you use this experience or research experience into sort of your future plan?
Yeah, so I can't account for all of the new generation of doctors or anything, but personally, if I was a doctor, I would want to keep my mind open to a lot of things, take into account culture, take into account, different alternatives, and not just be so closed-minded with just one route of Western medicine. And I feel like having these exposures and these experiences has definitely created who I am and how I view the medical world and who I want to be. Thank you.
Thank you so much. Oh, yeah. Thank you.
Stop share
Oh,
and then pull your presentation up. And then you can go back to your screen in Zoom. it yours?
Yeah.
Go ahead.
Do you know how I can get the bar off the screen? Is that possible? Might block some of my video. Thank you. Oh, perfect.
Oh, I didn't share yet.
Oh, okay.
There you go. This one?
Yeah.
I do not know how to get that. I can text to see if somebody can come back.
Just no, it's blocking some of the stuff on it.
Let me go see if I can get somebody to come in because I
Okay.
honestly don't know how to get.
If they can't in a couple minutes, I can just read it.
Okay. Let me see what I can do.
Oh, perfect. Thank you for your help. Yeah. She is she's here to talk to us about the chronic stress, how it elicits sex effects on the gene expression in the hippocampus Japanese families. Whenever you're ready. Perfect. Yeah. Thank you for that introduction. As Dr. Waldrop said, I'm Delilah, and I'm a senior biology major. And over the last year, I've been working on this project in the Lopez lab under the advice of Dr. Patricia Lopez. So what is stress? We define stress as any environmental or physical pressure that elicits a response from an organism, specifically that response being a disruption in homeostasis, that state of balance. And it's important to understand that there are differences in stress when stress is acute and temporary, lasting a short period of time. It generally elicits a protective effort on the body, so they're generally beneficial responses. That includes a boost of motivation or energy to get us out of whatever immediate situation we're faced with and immediate survival.
However, when that stress becomes chronic and long-lasting and continuous, the beneficial responses become increasingly detrimental on the body. So you can have a lowered ability to fight diseases and infection, an increased blood pressure, and then a negative effect on mood, sleep, and memory, which are also associated with a variety of really damaging diseases, PTSD, depression, Alzheimer's. So very negative effects on the body when that stress is chronic. And an especially interesting structure that we consider when thinking of stress is the hippocampus, a structure in the temporal lobe of the brain involved in learning and memory. And this structure is especially interesting because it is so highly susceptible to stress. It's densely concentrated with glucocorticoid receptors, so stress receptors, and it allows stress hormones to easily target and damage this structure. With that context in mind, we asked the question, how does chronic exposure to a stress hormone affect gene expression in the hippocampus? In getting into our experimental design, our study species were male and female Japanese quail. This is a species that can be chronically exposed to stress in captivity. They're used as poultry, so overcrowding, competition for resources, bad living environment, and then they can be chronically exposed to stress naturally during migration. And our stressor, our stress hormone was corticosterone, or CORT. It is the main avian stress hormone, and it's also equivalent to the main mammalian stress hormone cortisol, and is the main stress hormone in humans as well. So what we did was we had a treatment group of, or a total group of 20 birds, of which they were given their treatment through water. So if birds were stressed, they were given CORT laced in their drinking water. And if birds were not stressed, they were just given regular drinking water. And that was done for 5 each male and female control and CORT treatment. Afterwards, we did a brain dissection. We extracted their brains, flash froze them, coronally sectioned them, and took 100 micrometer slices, seen here. And then right here is the hippocampal region. And we took punches, hippocampal punches from that region, of which we did an RNA extraction from those samples and sent that off to be sequenced in order to retrieve our transcriptomes for our analysis. Afterwards, we performed a differential gene expression analysis using a bioinformatics pipeline from our sequencing data, specifically from a gene count file with approximately 21,000 genes. And that was performed, as I said, with a differentially expressed sequencing package in R and RStudio. And of that analysis, we extracted our significant upregulated and downregulated male and female differentially expressed genes. And when I say differentially expressed genes, I just mean genes that were responding to that stress or changing their expression due to that stress. And genes were considered DEGs or significant if they were less than a significance of 0.05. And finally, we performed a GO-term enrichment, a gene ontology enrichment to determine the biological pathways of those DEGs, which is how we look at gene function globally. And onto our results. So here we have the total number of differentially expressed genes in females on the left and males on the right. These are our volcano plots. And our DEGs are the genes in blue. So on our X, we have our log fold change, which just tells us if how genes were changing their expression. So if they were negative and they were downregulated, and if they were positive, they were upregulated. And then on our Y is our significance. And our top 10 genes are labeled as well as the overlapping genes. So this figure is just a summary of our DEGs here in blue. females and males. And our total number of DEGs in females was 113, and nearly half as many genes responding to stress in males, 64. And of those DEGs between males and females, only 5 were overlapping, which tells us that male and female quail are showing divergent genetic responses to chronic stress. So there are nearly twice as many female genes, they have a greater response to chronic stress, and their response is very different due to that very low amount of overlapping genes. And looking at the profile of our female differentially expressed genes, we saw that it was indicative of a sex gap in hippocampal stress responses due to nearly double the amount of female DEG. And looking at a specific example of a female DEG, this SSTR2, we can see on this slide, it was the most significant female DEG. And on our X, we have our treatment groups, and on our Y, we have our amount of gene expression, essentially. So we can see that this gene is upregulated in stressed females, and it also follows a similar pattern of upregulation in stressed males, but it wasn't significant in males. And this SSTR2, somatostatin receptor 2, is a gene involved in neuroendocrine function and is involved in the central nervous system as well. And some studies showed that when this gene was depleted, there were increased chronic stress-induced deficits, which was possibly telling us that it was so highly upregulated because the females were trying to counteract that negative response. The upregulation was some sort of protective effect from the negative stress responses. And what are our implications on the regulation of hippocampal structure and memory in the females? So here we have our gene ontology plots, as I said before, how we globally look at gene function. And these plots just tell us the biological processes that those the female DEGs are contributing to. So on the left, we have our upregulated female genes, and what these terms tell us is that there was a possible restructuring in the female hippocampus. And then on our downregulated female DEGs, these terms were telling us that there is a possible decrease in memory formation ability or a decrease in memory and learning. And so what are our main conclusions? Female and male quail show divergent genetic responses to chronic stress. The profiling of female DEGs is indicative of a sex gap in hippocampal stress responses, and female DEGs suggest a hippocampal restructuring and a possible memory formation disruption, as well as a disruption in learning. And then just some potential implications. Due to the difference in stress responses, amount of DEGs, the few overlapping genes, and what we learned from our female terms, the effects on hippocampal structure and decreased memory, it may be important to consider different therapeutic strategies for dealing with those stressors for men and women. And with that, I'd like to thank Chapman University and Schmid for funding my summer research, my faculty advisor, Dr. Patricia Lopez, for giving me this project and guiding me through the whole thing, and then Dr. DeBruin for animals and experimental setup, and then Dr. Iba for guidance through this process. And with that, I'll take any questions at this time.
Is it, so I know you said that the hormone being tested, cortisol and cortisol, are more or less analogous between these birds and humans. But is is the response likely to translate across different organisms? Or is it possible that that response is sort of hard encoded into how these specific animals might respond, and a rat or a human is going to have more moderately to more extreme?
That's not,
response?
that's not something I directly looked at, but considering they're essentially equivalent in mammalian species and avian species, I could predict that they have similar responses, but I can't. I don't know enough about it to give a better answer than that.
Can you hear me?
Yeah.
Yeah, so can I conclude that that's why men die early than women? Because we don't respond, we have to express.
I mean, studies do show that overall a lifetime of chronic stress can actually decrease your lifespan. But I guess with that in mind, you would think that females would die earlier if they have a greater response to that chronic stress. So I'm not super sure on
I
that.
don't see that reason that's all the women left, no men.
Yeah.
Hi, can you hear me?
Yeah.
Yeah, so, I'm not sure if this was addressed, maybe we haven't missed it, but, you mentioned that there were only 5, like, overlapping, like, regulated genes in
Yeah.
males and females. So for the ones that are kind of, like, not in that overlap, the ones that are different between those two groups, what are, like, their potential
we looked at the implications of those genes in the females. All of these plots are the processes that the female DEGs are contributing to. So we looked at that. We were thinking due to the much lower number of male differentially expressed genes, we couldn't obtain any of these plots at that significance just because there was a lower number. But I would think there's a similar stress response, but it's hard to say when those genes are different between the females and males. I don't know if that answers your question, but.
One in your group looked at other stressful agents to see the similar response.
Not that I know. My professor will know. And not that I know of.
What that stresses like diseases are stressors as well, but we haven't done comparisons of different types of disease stress.
If you looked at least some of the projects I looked on as far as disease, lipopolysaccharide, which is a component of bacteria that stimulates an immune response. But other than that, yeah, not that I've done. Yeah.
It's actually the same question first. So you mentioned that some of these restructuring changes might lead to reduced memory formation. Have you directly tested that in animals, and if not, what would you expect to happen if you did the same thing, even like a memory test or test?
Oh. I guess given what we know of the hippocampus, I would assuming these findings, I would think that they would have decreased learning and memory, but that's not something we tested. We just did the gene expression. That's an interesting question. Yes.
After graduation?
So I don't have a clear plan yet, but I'm hoping to go into the industry after graduation, possibly research, and then consider if I want to go back and do a PhD in biology. So, thank you. Yeah.
All right. Thank you, Lily. Couple minutes to switch over. Turn it to now here. No.
It's still in my email.
Oh, it's still in your kid?
Oh, I see that.
Okay, let me just sign it all.
Yes.
Okay, thank you.
And you're the last, so please.
Oh, wait, did it work? Oh,
Yeah.
I guess.
Did you share the Zoom? Bring it back over. There you go.
Thank you so much.
Now you can probably expand your screen. Here. You want to hide. I don't know if he's sure.
Oh. That's okay, I can just do right.
Are you good?
Yeah. Is there a way to remove that?
Thank you. Have to download it.
Oh, okay. Then.
This one, right?
Okay.
There you go. And then those things. Let me make sure you're sharing now on my screen.
Okay.
influences.
Right, you're gonna move it. Okay.
Being in there with us for a couple technical issues, but we're all ready to go now. So our next speaker is Chathuni Liyanage, and she is in Dr. Lopez's lab as well. She will be talking about a treatment with bacterial lipopolysaccharide. Downregulates non-bacterial immune response.
Thank you for the introduction. So as Dr. Waldrop said, my name is Chathuni Liyanage. I'm a senior undergraduate student, and I've been conducting research with Dr. Lopez for the past couple of years. And the title of my presentation is Lipopolysaccharide Downregulates Non-Bacterial Immune Responses. So to give you a brief introduction, we were interested in seeing how our bodies respond to infection and also how it adapts and how these adaptations can lead to different changes on the different molecular levels, whether they be behavioral, genomic, or physiological. And essentially, we get an infection when some type of microorganism invades our body and is able to cause potential harm to the host. And so once our body detects the presence of one of these foreign entities, it elicits a cascade of responses in order to both defend the body against infection and also to protect the host cells from potential destruction. So essentially, our immune system is what provides us this protection. And depending on the type of pathogen that we are exposed to, the immune system will elicit different types of signaling cascades and different types of responses in order to best tackle the specific pathogen. For example, bacterial pathogens respond best and are more susceptible to destruction by what are known as complement proteins, whereas viral pathogens are actually more susceptible to damage by what are known as interferons or cytotoxic T cells. And so essentially, the importance of seeing how the molecular mechanisms are changed or are activated during an infection response is important in order to aid in the development of targeted therapies. So we established this model in the Japanese quail as they've been seen to be an ideal model species in studying disease. Lipopolysaccharide, E. coli lipopolysaccharide, or LPS, is the agent that we use to induce an immune response and is a common agent used by many researchers in order to study infection responses. Essentially, it is a component of the gram-negative bacteria cell membrane, and it is also a common antigen that is responsible for many of the bacterial infections that we as humans face. So we decided to look at the blood transcriptome or the blood genomic response of our individuals in order to get a global picture of how infection causes disease at that level and how we were able to quantify the different genetic changes that occurred from infection. Now, this leads us to our question of how does an immune threat affect the behavioral and blood genomic responses in Japanese quail? So to give you a little rundown of our experimental design, we had a treatment group and a control group. Our treatment group was injected with LPS, whereas our control group was not injected with anything, including a sham injection or a saline injection. And this is in order to prevent introducing additional variables that could come about through an inflammatory response at the site. So after injection, all individuals were moved to their observational cages where they were left for the duration of 3 hours. And throughout this time, they were recorded remotely using security cameras in order to conduct a behavioral analysis in the future. And at the end of this 3 hours, we collected their blood samples and performed an RNA extraction followed by a subsequent purification. And the purified RNA was sent for sequencing, to which we obtained a gene counts file in order to do a gene expression analysis, which was done using a bioinformatics pipeline on our studio. And next, we did a functional analysis, which I will get into a little bit later in our presentation. And finally, a behavior analysis was done in order to quantify the different types of sickness behaviors. And this was done using a software called Boris, which essentially we were able to quantify the amounts or occurrences of either eating or drinking behaviors and standing duration. So what is a sickness behavior? Essentially, when an organism is sick, they tend to have a lot of different behavioral changes that are implemented, whether they are decreased eating and drinking, or just overall increase in rest. Essentially, these are just adaptive behavioral changes that occur when there's an onset of infection. So this is an example of how we quantified our sickness behaviors. This one is eating. And as you can see in the control group, which is the red, and then our treatment group in the blue, there's a decrease in the number of eating events that we see in our treatment group. And this is consistent with just an overall decrease in appetite when you are infected or just feeling sick. And this same behavior is seen in our drinking events, which you can see that there is quite a fewer drinking events in our treatment individuals. And finally, with standing, this is probably the most significant difference that we could see between the two. And seeing that though there was a longer duration of time that the control individuals spent standing compared to our LPS, we can see that this is pretty consistent with how we feel when we're sick, which is overall fatigue and lethargy, along with just a total lack of appetite. So after our behavioral analysis, we also conducted a differential gene expression analysis, and this is our volcano plot, which essentially shows us a general distribution of the genes which were differentially expressed between our treatment and our control individuals, and the ones in the blue up here are the ones that were significant. And of the 20,000 plus genes that we had sequenced, a little bit over 620 of them were differentially expressed between our treatment and control group. And I will be referring to the differential expressed genes as DEGs from now on. So to the left, we can see that these are the genes that were differentially downregulated. And to the right, we had around a little over 200 that were differentially upregulated. So from our gene expression analysis, we were able to conduct a functional analysis, and essentially what we were able to do is cluster the different genes that we saw were expressed significantly by their function, and it gave us an overarching picture of what processes these genes were involved in. And these are the upregulated pathways that our genes were involved in, and there was nothing too surprising here. We expected a both general and specific immune response in response to the LPS bacterial immune threat. And so we can see that there's things such as lymphocyte and leukocyte cell differentiation and activation. And again, that just goes along to show us that there was some sort of global immunoprotective response that was elicited in response to our immune threat. And so next I'm going to get into some of the downregulated pathways. This is where we saw some things that were a little more surprising. And before I get into the nitty gritty, we just saw that there was an overall downregulation of non-specific responses and non-bacterial specific responses, which in terms of a trade-off makes sense, because in order to allocate energy to where they're more prominently needed, such as for producing the immune cells we need to tackle infection, we would expect that these non-specific responses would be downregulated. But what was really interesting to us is that we saw that there was something called a interferon type 1. A lot of the genes were related to the production or regulation of these that molecule, and essentially, given its importance in the infection response, we were a little bit surprised that it was so significantly downregulated. However, upon further analysis, we were able to see that it did play a pretty differential role between bacterial and viral infections. In viral infections, it is in charge of inducing the production of antiviral molecules. So it would be expected to be upregulated in that case. However, in a bacterial infection, such as the one that is induced by LPS, it's actually only needed in low levels in order to elicit an initial response and activate the cell-mediated response. And actually at high levels, it can cause immunosuppressive effects. So that was really interesting to see that it was relatively downregulated in our bacterial cells. So one of the main things that we took away from this is that there were a significant number of genes involved in pathways that were involved in viral defense and responses. And what this told us is that in order to have enough energy and enough resources to combat the bacterial pathogen, our bodies were reallocating resources from non-specific pathogens such as a virus and allocating them where they're more needed in order to maximize the infection response. And so that was what kind of indicated us that there was some sort of trade-off between non-pathogen specific and non-specific responses in order to really just maximize the effect. And so this brings us to our conclusion. Based on our behavioral and genetic analysis, we can see that LPS did in fact reconstitute a model infection, and that the genomic changes show that there is a broad upregulation in both innate and adaptive, or specific and non-specific immune responses. And there was a selected downregulation of non-specific or non-pathogen specific defenses and responses. And what this shows us is that in the global response to infection, we can see that there is a dependency on the balancing of resources in order to cater to the specific pathogen or immune threat that is available. And what this tells us is there is a potential energetic or physiological trade-off that's implemented in order to ration the limited resources and energy that our body has in order to ensure the recovery of the host cell. And so I would like to thank Dr. Lopez for continuous support through my undergraduate career and for giving me all these opportunities in research. And I would like to also thank Dean Iba for his help throughout the semester and for always encouraging me during these presentations. And of course, my parents for their endless love and support and for everything that they've given me. So thank you.
Can you give me some example of LPS?
Yeah, sorry, let me go back.
Is it in food or?
So it's a component of gram-negative bacteria, which is a type of bacteria, and it's found in their cell membrane. And it's essentially a part of the cell membrane that is what our body recognizes as the immune threat. And so this is right here. It's this portion, this whole thing. And this is the actual cell membrane, but we just use this part because instead of using a live pathogen that could have made the, if we actually got them sick using a live pathogen, it would make it transmissible to the other individuals. So by using this, we were able to induce an infection without actually getting our individual sick.
Why did you use the Japanese
So in our lab, Dr. Lopez has been using Japanese quail as our model species, and also just for this project in particular, there have been a lot of studies showing that they are an ideal species in studying infection and disease. So that's why in this one it's relevant.
To expand on that, I'm curious to see would you possibly want to do this in invertebrates because they only have the innate immune system and see how they react.
Yeah, that would definitely be an interesting thing to see. I think since we did it based off of what was most similar to a human infection model, which is why we used the Japanese quail. But I think to also just see how the innate immune response solely works would definitely be something that we could branch off on. So I will be applying to dental school this cycle, but I would hope to continue being involved in research and potentially seeing if that takes me elsewhere career-wise. Thank you.
Session. Let's give one more round of applause for our great speaker.